So that is the first part, introduction and application of various molecular modeling tools in the process of drug discovery. And second part is of a small case study with special emphasis on anti-leishmanial drug discovery, what we have done. So with this small brief introduction, I will be starting. So first, all of us might be aware what is in the drug discovery. Okay, it's nothing but the developing of a new drug from original idea to the launch of a finished product in a complex process, which can take a minimum of 12 to 15 years with the estimated expenditure of 1 billion. Okay, so then the idea for a target can come from a variety of sources, including academic as well as clinical research, as well as from the commercial sector. Yes. So in order to do that, it may take many years to build up a body of supporting evidence before selecting a target for the costly drug discovery program. So in the right hand side, the figure what we are showing is a simple drug discovery process. So nowadays it can be represented in various ways. So this is one way of representing the entire drug discovery process. So all of us might be knowing it will start from the identification of disease. So based upon the requirement or based upon this VRIT or based upon the demand. So the segment identification will take place, that is identification of disease. Then once a disease has been identified, the second immediate process comes in place is isolate protein involved in the disease. Is nothing but target isolation, separation, purification, as well as target identification, And second, once a target has been identified, means it needs to be validated. So that is target validation. So this is this part. Okay, so this part of our process is called target discovery. So that is target discovery followed by lead discovery. So these two are all the important phases of drug discovery, wherein which various types of molecular modeling tools can be used. Okay, so in order to shorten the duration as well as in order to reduce the cost or price involved in this new drug discovery process. Okay, so these are all the regular routine process. So all of us might be knowing. So this is that thing. What I am talking is this thing only. So initially target identification, validation, and followed by lead identification and optimization. So once lead has been optimized, then it will go to preclinical development in animal studies. Then it will enter into the clinical developmental phase, which consists of 4 phases. That is phase 1, phase 2, phase 3, phase 4. Then after that, it will come into the market. That is post-marketing surveillance. All those things it will come. Okay, so these are all the various phases and stages of drug discovery. So that is discovery and development, preclinical research, then clinical development, then FDA review as well as getting approval, then post-marketing monitoring, everything. So these are all the new strategy for drug discovery. So far, what I discussed is earlier ways of drug discovery. So now this is the new strategy for drug discovery. So in this, 2 things has been incorporated. One is improvement of efficacy and prognostic profiling in order to check for tractability. Then second, far most important thing is risk assessment and clinical development. So these 2 things nowadays has been incorporated. So these 2 things needs to go hand in hand in order to reduce the drug failure at the end. So everyone might be studied. So for so many years, several drug discovery processes are being taken place, but the number of molecules which appear in the market, the ratio, if you see, it is very less. Okay, so if we start to analyze what are all the various reasons for this, so in that main 2 things is a toxic effect as well as non-trackability as well as this risk side effects are this thing. So in order to avoid that thing, only these 2 parameters have been incorporated in the recent drug discovery process. So in order to take care of this thing only nowadays, at our academic level itself, we are predicting this trackability profile like ADMET, in silico ADMET predictions as well as toxicity prediction kind of thing. Okay, so these are all the various phases of CAD, which is mainly which is involved in the drug discovery process. So two things I told. One is initial phase of target discovery is fully going to discuss only about target identification followed by target validation, and second phase is only going to talk about lead discovery. So that is hit identification, then lead identification followed by lead optimization. Okay, so as far as your target discovery is concerned, that is for target identification as well as target validation. So there is no much computational tools has been used. So your real experimentation techniques are basically used, like X-ray crystallographic studies or NMR studies, or recently some kind of de novo design or homology modeling kind of things are involved in the target identification validation. And majority of our computational tools are mainly involved in this second phase, that is lead discovery. So in that also two phases. One is the lead identification, that is hit and lead identification followed by lead optimization. So if you see lead identification, so what are all the things involved is database filtering method, then basic alignment, then similarity searches, then bioforce, then virtual high throughput screening, then diversity oriented selections are the techniques or tools involved in the lead identification process, then followed by lead optimization. So for this, the various tools generally used are QSAR, that is quantitative structure activity relationship, and its recent versions like HQSAR, that is 3D, 4D, 5D, all those things. Then ADMETSAR, that is absorption, distribution, metabolism, excretion, and toxicity prediction. Then followed by combinatorial library synthesis, then de novo design. So all those things are mainly focused on lead optimization techniques. Okay, so these are all the various phases of techniques mainly involved in the drug discovery as well as the lead discovery phases. Then these are all the basic modeling strategies. So as far as our this thing is concerned, our that is lead discovery is divided into two. One is ligand-based drug discovery, and another one is structure-based drug discovery, that is LBDD and SBDD. So based upon this, when and how to use this LBDD and when and how to use this SBDD is mainly you can see from this table. So for example, how to interpret this table is like this. So for example, whenever ligand structure, okay, so ligand means a small organic molecule or simple peptide or nucleosides kind of thing. So whenever your ligand structure is unknown as well as your receptor structure is unknown, means then we need to use this strategy like high throughput screening, combinatorial chemistry, this thing. Then for example, if your ligand structure is unknown but our receptor structure is known, then either we need to use de novo design or visual screening. Okay. Then second, third category is whenever our ligand structure is known but our receptor structure is unknown, means either we can go for this 2D, 3D QSI and pharmacophoric modeling kind of thing. Then whenever our ligand structure is known as well as receptor structure, receptor means our target structure is known, means at that time either we can go to molecular docking kind of thing. So these are all the basic selection molecular modeling modalities or strategies. So these are all in detail specific to hit and lead identification strategy. So here that first step is hit identification in order to identify the hit. So these are all the various strategies being widely used. So starting from here, that is virtual screening, docking, similarity search, de novo, scaffold hopping approach. Then apart from that, fragment based screening, that is FBDD. Then building blocks based, that is ROT, RO2 or FSP3. Then high throughput screening, then phenotype screening, all will result in the identification of hit. Once hit has been identified, then it needs to be optimized. So this is about docking. So docking is nothing but finding out the best match. Okay, so molecular docking is a type of bioinformatic modeling which involves the interaction of two or more molecules to give the stable adduct. So depending upon the binding properties of ligand and target, it predicts the three-dimensional structure of any complex. So this is the diagrammatic representation of your docking. So these are all the things is happening. So this is our target, I mean either receptor or protein or enzymes, whatever it may be. And this is our ligand. So ligand will be either any small organic molecule or simple peptide or something. So by means of docking process, so we are finding out the best match. So how this ligand is interacting or how this ligand is uh, binding or shows tight complex in the active site of our target, all those things. So based upon its binding or based upon its docking pose or based upon its binding affinity, so we will get the docking score. So that results, that docking studies results will be represented in terms of docking score. So docking score is nothing but the binding free energy. Okay, so this is the thing. So then if you want to prove this thing, which one is best docked pose, then we need to go for X-ray structure as well as delta G, that is difference in the binding free energy calculations. So this is the process mainly involved in the this thing. So in this docking, there are two things are involved. One is docking pose and one is docking affinity. So docking pose is nothing but the we can compare between the same molecule. So same molecule one, if you are taking so how that one molecule is interacting in a various poses. So that is docked pose. And second one is binding affinity. So binding affinity is mainly used to compare between molecules. For example, let us say we have designed 50 molecules. So out of that 50 molecules, which molecule showed better affinity? So that will be determined based upon the docking affinity kind of thing. So these two things are involved. So in order to execute this operations or process, there are three things are involved. One is algorithm, that is a docking algorithm, is nothing but a search algorithm. Then second one is scoring function. Okay, and third one is post docking analysis tool. So these three things are involved in order to execute this process of docking. So this docking in general, it generates different possible adduct structures that are ranked and grouped together using a scoring function in the software. This docking simulation predict optimized docked conformer based upon total free energy of binding of the various systems. So these are all the other applications of molecular docking. So by means of doing molecular docking, so mainly we can perform this ligand target binding rationalization by means of identification of the structural determinants, which is necessary for the efficient ligand receptor binding. So apart from that, it can also be used for virtual screening. So virtual screening is nothing but identification of a most appropriate compounds or ligand from a bunch or from a bulk of available databases, either inbuilt or is freely available or paid. This thing which will readily modulate the disease-related targets and their optimization, and that it can also be used for target fishing and profiling. Target fishing is nothing but, for example, we have active ligand, but we don't know the exact target, how it is showing activity. So in that case, we need to perform this target fishing is nothing but prospective molecular target identification. Is nothing but prediction of targets for the active compounds on the basis of ligand receptor complementarity approach. So apart from this, this molecular docking can also be used in the prediction of adverse drug reactions for our active molecules based upon the prediction and the rationalization of drug off-target activities based on the complementarity between ligands and target. And apart from that, this molecular docking can also be used in the area of polypharmacology. So polypharmacology is nothing but identification and optimization of compounds that simultaneously modulate a set of more than one targets, major targets involved in the same disease or disorder in the maintenance. Then at last, it can also be used in the one of the very much emerging area of drug repurposing or drug repositioning. So everyone is aware. So it is one of the emerging and widely used approach in this pandemic situation. That is nothing but identification of a novel therapeutic relevant targets for already marketed drugs and known chemical and natural entities. So these are all the various applications of molecular docking. And apart from that, this also can be used in the area of AI, that is artificial intelligence, deep learning, machine learning, and statistical methods. then binding free energy methods, then molecular dynamics, as well as ligand-based approaches. So in all the things, post-docking screening and pre-docking screening is possible. So this is a few glimpses related to drug repurposing. So in our case studies also, we are going to highlight these drugs are repurposed for leishmanial therapy kind of thing. So these are all the workflow of our basic this thing. It's a strategy to identify new therapeutic uses for marketed drugs or discontinued or self drugs and drug candidates currently in clinical development. Although not a recent concept, so this is a very old concept, but now is emerging. So has a gained momentum over the past few years, and several drugs has been successfully repurposed. So this is mainly in order to reduce the cost as well as reduce the duration in the invention of already existing drug for a new this thing. So these are all the some statistical report regarding drugs repurposed for different therapeutic area is almost 60%. Then drugs repurposed for same therapeutic area is almost 40%. So in this, majority of onco neuro drugs are mainly involved. So this color code indicates that thing only. So this one, these two are major, that is so oncology and neurological symptoms. So these are all the major in both different therapeutic area as well as the same therapeutic area. So this oncology and neurological indications plays a vital role. So these are all the few successful drugs that has been repurposed. I mean, very few lists only I am showing. So full list will go several. So all those lists I am not showing. These are all the very few structures that has been successfully repurposed by using this. Then second important tool, what I told is virtual screening. So virtual screening is nothing but a computational tools mainly to narrow down, okay, mainly to narrow down or mainly to identify hits from a pool of larger databases. Basically, the computational technique mainly used in the drug discovery to search libraries of small molecules in order to identify those structures which are most likely to bind to a target, typically a protein, receptor, or enzyme. So that is the thing. So this is the diagrammatic representation of a virtual screening. So these are all the database compound, and this is our target. So we need to define. So after defining, so we can incorporate some filters like drug-like or lead-like filters, as well as the toxicity filter, bad apple or bad process, then pains, as well as ADMET filters. So after applying these filters, so from this library of compounds which passes through this filter only will come as a hit. Okay, so that hit we need to perform molecular docking in order to give a ranking kind of thing. So this is the entire workflow. So this slide will indicate the influence or impact of molecular docking in recent publications. So if you see in the recent years, so this is 2013 and this is 2018 and this is 2019. So in recent years, especially 2017 to 19, in this past 3 years, see the impact of molecular docking in the publication. It means the number of published research articles with molecular docking studies is increased day by day several fold. So that's what this simple statistical bar diagram is informing. So that is the thing. So up to this were basic approaches of various molecular modeling tools involved in drug discovery. Then our own research findings, just a few slides I will be discussing. That is, I told you. So currently we are working in one of the allied opportunistic infections of AIDS, that is leishmaniasis. Is a protozoal diseases. Comes under the category of neglected tropical disorders as per the WHO World Health Organization definition. So it's a vector-borne neglected tropical disease mainly caused by an applicate intracellular parasite of a protozoa of a genus called leishmaniasis in the order of Kinetoplastid, family Trypanosomatidae. So this leishmaniasis is mainly broadly classified into three. One is visceral leishmaniasis, then mucocutaneous leishmaniasis, then cutaneous leishmaniasis. So out of this cutaneous leishmaniasis is self-curable, and that remaining that visceral leishmaniasis is the very harsh form. So mainly it will affect the visceral organs like liver, lungs, and heart kind of thing. So then in between that mucocutaneous leishmaniasis mainly affects the various mucous membranes of our various body organs and tissues. So in humans, it is transmitted mainly by sandfly species of Pleopotamus. So this is the diagrammatic representation of the sandflies. So these are all the fact sheet. So how and where? So these are all the main areas. That is one of them, that is India and South Africa is the major vulnerable. So this is the statistical data. So people around the world is this many new cases every year. This many. So these are all as per World Health Organization 2019 data. So these are all the various drugs repurposed for leishmaniasis, starting from all drug category we have, that is metformin, mainly used for type 2 diabetes, then lamivudine, the antiviral drug, then third one is cladribine, that is anti-cancer again, then tenofovir, again it's antiviral, then perphenazine is anti-psychiatric drug. And these are all the current workflow what we have followed. So we have started with virtual screening by taking a zinc database. Then we got a hit of around 1,000 molecules from around 50,000 molecules, 1,500 molecules we got. Then all those 1,500 molecules we have subjected to SP docking as well as XP docking. Then we got hit of around 130 molecules. So out of that 130 molecules here, I am highlighting only top 5 molecules. Okay, so this is the general workflow. So these are all the things. So these are all the hardware and software configuration what we have used. Computer configuration is done. Then these are all the drawing tool what we have used. Then major software what we have used is Schrödinger suite. That's in 2019 one. So the database we have used for our study is a zinc database. It's a freely available database. Then protein target or what we have used for our current study is 2JK6. It's nothing but leishmanial tryptophan reductase. So by using this minimal information, just we have started. So first we have prepared our protein by using the protein preparation wizard of Schrödinger glide. Then we have prepared the filtered ligands by using ligand preparation wizard of this Schrödinger glide. Then in that we have used HTVS as well as ASP standard precision and extra precision docking module used for filtration of ligands. Then finally we have used a XP visualizer for the viewing of the generated pose of the top 5 hits. Then finally that top 1 molecule we have performed molecular dynamics by using the Desmond. So Desmond module of the Schrödinger glide. So these are all the hardware as well as software information, and these are all the information related to zinc databases. So total molecules available in this FDA approved drugs only we have considered. We have not considered new chemical entity which is involved in this zinc database only. We have taken FDA approved drugs. So that is around 4,500 molecules. Then initially we have applied a filter, ADMET filters. So in that we got only 3,700 molecules. Then we have applied HTVS. So in that some more drug molecules got filtered out. So at the end we got only 2,600 molecules. Then all these 2,600 molecules we have involved, we have performed a standard precision mode of docking. So in that we got docked pose only for 1,400 molecules. And that 1,400 molecules we have subjected to extra precision mode of docking. So at the end of this extra precision mode of docking, we got only 800 molecules got docked pose. So from that 800 molecules, we analyzed only top 10 or top 20 molecules only. So in that entire study, we put a docking score cutoff value of -5. So ultimately, we got only 400 molecules. So if we can increase this docking score slightly increase slightly to -7 or -8, means then this number of molecules again will further go down. Okay, so this cutoff value we can decide based upon the docking validation part. It's nothing but how much docking score we are getting for the co-crystallized ligand upon our docking validation, that is extraction and redocking process. So based upon that, we can put cutoff value. So in our study for co-crystallized ligand, generally we got docking score of -14 at the time of docking validation. So we can keep -10 also, but in that case, we analyzed only double digit we got the hits around 75 or 70 molecules only we got. So we thought that 75 or 70 is very less in numbers from 4,500 molecules. So that we have reduced the docking score cutoff value of -10 to -5. So we got further increase, that is around 70 hits has been increased to 450 molecules. So we stopped this. We have not further reduced. So these are all the things. So these are all the diagrammatic representation of the workflow what we have followed. So I told you. So initially we have prepared the protein. So that is 2 JK6 is nothing but leishmanial tryptophan reductase. So again, this selection of these 2 JK6 is also very important, critical. So for that we have applied 3 or 4 parameters. One is resolution. In and around 2 resolution is prescribed or advisable, and second one is that IC50 value of the co-crystallized ligand, which is present in this protein, that is 2 kg. That is second thing. And third one is R free value as well as B factor. So we have considered. So these 4 are the selection criteria. So based upon that, we have chosen this 2 JK6. Then after selecting this, we have prepared this 2 JK6 as well as we have prepared this ligand, which is in this FDA approved thing. Then we generated a receptor generation by using that protein preparation wizard only. So once ligand as well as protein as well as split has been generated, means we have launched that HTVS, that is a high throughput visual screening. So half of the things got filtered and we got only around 2015 molecules. Then we performed molecular docking. Then we got the hit. Then that hit we performed the molecular dynamics. So this is the simple workflow we have followed, and this is the validation part. So I told you. So this is very important as far as your docking is concerned. So this validation we have performed after doing extraction of the co-crystallized ligand. Then we prepared that ligand. Then again we redocked. After redock, then we have superimposed that redocked pose with that co-crystallized pose. So after doing this superimposition, then we have measured the differences between this docked pose to the top of the co-crystallized pose. The difference needs to come less than 2 Armstrong. So exactly we got around 2 Armstrong only here. So that we can ensure that the validation or the docking protocol what we have developed can be fit for further usage of our developed or further usage of our shortlisted hit molecule. So this indicates the docking validation, and these are all the docking integration of top hits. Docking integration is nothing but the docking interaction. So I told you. So this is the co-crystallized ligand. So co-crystallized ligand docking score is -14. So when compared to that, our top hit molecules docked score is -14. So these are all the various interactions. So mainly, so all of us might be knowing. So docking. So what are the various factors or various forces influence the docking interactions? Among those, first and foremost thing is hydrogen bond, then hydrophobic interaction, then pi-pi interaction, then salt bridges, then aromatic interaction. Everything will come. So in that, so we are focused on hydrogen bond interaction. So mainly this hydrogen bond interaction, this 1, 2, 3, 4, 5, 6, 7, 8, this 8 amino acid residues, which is a part of our active site residues of these 2 JK6 are involved. So if we compare these residues, so it is almost similar to that of the amino acid residues, which is involved in the hydrogen bond interaction with the co-crystallized ligand. So because of this similarity in the interaction, I mean the amino acid residues involved in the hydrogen bond interaction to that of the co-crystallized ligand, the docking score of our hit will be almost comparable equally. so minus 14.7 as well as minus 14.60. Then that top hit molecule was identified as a 5-methoxy tetrahydrofolic acid. So all of us might be heard about this folic acid. It's nothing but biologically active form of a synthetic vitamin B9, that is folic acid, is mainly used to treat anemia caused by folate deficiency. Then the second molecule, top 2 hit molecule, what we got is that is leucovorin. So it's nothing but the levo form, levo enantiomer of leucovorin. So this leucovorin is again is nothing but mainly used as a anti-cancer agent. Combination therapy in the colorectal cancer treatment along with 5-fluorouracil. Okay, so we have identified this leucovorin as a top second hit, and the top third hit what we have identified is the pralatrexate. So this is also again anti-cancer agent mainly used for the treatment of blood cancer. So it belongs to the category of anti-metabolite for the treatment of relapsed or refractory peripheral T lymphoma, is nothing but blood cancer. So then fourth compound we have identified is this chlorhexidine. So it's nothing but disinfectant and antiseptic mainly. Then fifth drug what we have got fifth hit is labetalol. Is nothing but beta blocker, mainly uses antihypertensive as well as treatment of angina pectoris. Okay, so these are only 5 top hit. So we have analyzed in detail. So in all these top 5 hit, we got hydrogen bond interaction of almost 4 to 8. So out of that, first top hit molecule we got already 8. Then top 2 molecule also we got 8 hydrogen bond interaction. Then top 3, top 4, top 5 we got almost 3 or 4 hydrogen bond interaction. So it's clear this, if you see the docking score, so gradually it has start reducing. So minus 14 is reduced to minus 12, then minus 12 is reduced to minus 11, then minus 11 is reduced to minus 9. So this is minus 9, typographical error. Then minus 8, then this is co-crystallized Ligand. So these are all the docking interactions exhibited by the top 5 hit molecules. Then once this docking has been done, then we have performed molecular dynamics. So these are all the structures of the top 5 hits. Just now I have explained, that is 5-methoxy tetrahydrofolic acid, then leucovorin, then pralatrexate, then chlorhexidine, then labetalol. So these are all the top 5 hits. So currently we are in the process. So all are fortunately marketly available drugs. So we got the gift sample of a bulk drug of these 5, and just we are in the process of in vitro screening in the leishmanial strain. So both pronosticotes as well as amasticotes may just we are checking. So once if it is done, then we will be going into the in vivo model of amasticote, axonic amasticote form. So that thing else, those things are also under pipeline. So all these are all unpublished data. So just we are waiting for that in vitro as well as in vivo study result. So once it been correlated, then you'll be looking for the publication. So these are all the 2D and 3D docked pose interaction or representation of the top 1 hit molecule, that is 5-methoxy tetrahydrofolic acid in the active site of the target protein, that is 2JK6, is nothing but leishmanial 2000 detectase. Then I told you, so last phase of our computation study is this molecular dynamics. So this molecular dynamics, all of you might be aware, is nothing but simulations, which is mainly evolved into a mature technique that has been used effectively to understand the macromolecular to function relationship. So present simulation times are close to biologically relevant one. So currently we have performed this molecular dynamics for 5 nanoseconds. So now we are in the process of scaling up also in this molecular dynamics. So that 5 nanosecond study is only pilot studies. So once if we feel within this 5 nanosecond time, if our docked pose of our protein ligand complex is stable, then only we will be preferring into the scale-up mode for minimum of 100 to 150 nanoseconds. So because it needs a high computing power as well as time. So इसलिए just every molecular dynamic studies generally in first go we will perform at a small scale, that is 5 nanoseconds. So fortunately we got a positive response. So within that 5 nanoseconds, that top one hit molecule ka receptor ligand complex is quite stable. Then now we are in the scaling of process of also this in this. This molecular dynamics provides a detailed atomic or molecular level interactions. So these are all our findings. And prior to that, these are all the system builder parameter what we have used for our current molecular dynamics study. So these are all the grid, and these are all the ensemble NPT ensembles we have used, and we have used time for 1200 picoseconds or 5 nanoseconds. Then total atoms involved is this thing, that is 6,54,344 atoms are involved. Then water molecules involved is 12,720. Then temperature we have used is 300 Kelvin. Then grid box size is 10 into 10 into 10, that is of X, Y, Z axis. Then sodium ions 42, then chlorine ions 35, in order to maintain the charges of both macromolecule as well as small organic molecule in our study. So these are our research findings. So initially we have validated our target ligand complex by means of docking. So same thing we have replicated here to validate the protein ligand complex of the target compound, as well as to measure the consistency of ligand binding in the active site of the selected target. Molecular dynamic simulation study was conducted for the top hit first molecule, that is 5-methoxytetrahydrofolic acid. Then in this molecular dynamics, the complex was initially immersed in an anthropic box of species, that is a single point contact water molecules, and the solvent system was neutralized by adding this sodium. Then to equilibrate the system, the solutes were subjected to the NPT ensamples. Finally, the full system was subjected to an MD simulation, and for 5 nanoseconds at 300 Kelvin temperature and 1 bar pressure. The obtained simulation results were analyzed using the backbone Rmsd as well as Rmsf values. That graph or bar diagram also I will show after this. Then the root mean square deviation and the fluctuation, that is Rmsd and Rmsf, which describes about the backbone fluctuations of the protein ligand complex. So in this study, the Rmsd of our current MD simulation was found at 2 Armstrong as Rmsd and 3.7 lesser than 4 Armstrong for Rmsf fluctuations we found, indicated that this protein ligand complex is stable throughout the simulation time. Then this thing only I told. So we found stable in this small scale. For that, now we have scaled up to 150 to 200 nanoseconds. Then the protein ligand interactions found more hydrogen bonds, which makes stable the complex. So what are all the amino acids residues involved in the majority of the hydrogen bond interactions we found through molecular docking? Same residues we found in the molecular dynamic simulation also involved in the hydrogen bond interaction. So that similarity also is well matched in our results. Then the timeline and ligand contact clearly describes that the top molecule connected strongly to the surrounded amino acid residues and bottom molecules up to 99%. So these are all the results I am telling. So I mean that first one is of Rmst plot, and second one is of Rmsf plot. Rmst is nothing but root mean square deviation, and Rmsf is nothing but root mean square fluctuation. So in that first one, that Rmsd plot, so that time limit what I am telling. So this uppeak only I am telling, that is up to 2 or 2.1 Armstrong, and this top is 2.7 or something Armstrong. So this is the major variation. Remaining all are varied within 1.75 only. Okay, so in this, that red indicates the backbone, and this magenta indicates the alpha helix of our target, and this is the Rmsf plot. So here also that maximum fluctuation takes place at the 2.7 only, so it has not exceeded 4. So up to 5 nanoseconds it is permitted. So permitted in the sense it indicates the study population, that is the receptor as well as ligand complex are quite stable. So that is the indication. So it has not violated or it has not exceeded the prescribed limit of 4 or 5 Armstrong means. Okay. So this is the protein ligand contacts. So contact in the sense I told you, majorly your ligand receptor complex stability is enabled by means of various interactions I told you. So in that majority is, see, if you see that green indicates hydrogen and this blue indicates water bridges. So that thing only I was telling. So these are all the various amino acids into various amino acids. So majority of the amino acids involved are this thing. So it showed as the same hydrogen bond interaction with our protein ligand, and remaining this blue indicates that water bridges. So unfortunately here there is no ionic interactions and very minimal of hydrophobic interactions. So this thing can be confirmed from our talked this thing. See, majority of interactions are exhibited by means of hydrogen, and minimum only that is hydrophobic, that is aromatic, and very rarely only pi kind of thing. So that thing only is being replicated here. So from this protein ligand amino acid contact plot, we could able to verify majority of the interactions exhibited by means of hydrogen bond interaction by means of the active site amino acid residues are same is representing here as well in the molecular dynamic study. So these correlations only we could able to find from this plot. So these are all the timeline representation as well as ligand protein contacts. So see this. So this is the ligand protein contacts. So if you see, so this blue color indicates that is water bridges. See this. So this water bridges, that is ASN 179. Generally involved 77, and here also one that threonine 51 is also involved in the water bridges, and here that threonine 160 also involved. So apart from that, remaining hydrophobic as well as charged interactions also there, and this is the timeline representation. So in this x-axis timeline, so up to pi nanosecond, and y-axis that various amino acids involved. So in this two color representations, this dark color so indicates tight interaction exhibited by this 351 amino acid residues throughout our molecular dynamic studies, that is 0 to 5 nanoseconds. Likewise, here also this one. Okay, so this indicates that the ASN 179 amino acid residues involved in the tight interaction between this 2 to 3.5 nanoseconds only, and before that and after that it has not involved. So likewise, these two dark line indicates these amino acid residues, that is arginine 287 as well as this ASP 327 is mainly involved in the tight interaction with amino acid residues in between this 3.5 to 5 nanoseconds. So prior to that, it is not involved. I mean, not involved in the sense it's not involved in the tight interaction, but is involved in the mild interaction. So this dim color indicates mild interaction. That's all. This white color indicates it's not involved in the interaction. So these are all the inferences which we could mainly able to obtain from this timeline representation of the molecular dynamics results. So that's all. So of our glimpses of our findings. So this is the final summary. So over the last 4 decades, molecular docking as well as virtual screening has improved quite remarkably, contributing to the enhancement and improvement of pharmacology in addition to many areas of applied and molecular biological techniques. And the increasing demand for molecular dynamics, that one graph recent literature only depicted that every recent year, that is 2015, 16, 17, 18, the number of publication has been increased tremendously in a several fold. The revolution advancement of the technological background. So here, similarly in our case study also, the results reveals that the identified hits from virtual screening may act against this leishmanial species. So for that only I told further in vitro and in vivo evaluation of these compounds will be required, which are in the pipeline, in order to conclude that they will be effective for the leishmanial therapy. So these are all our final summary. So these are all the various references I would like to quote in order to make this material PPT. So these are all our recent research findings in this area. And finally, I would like to acknowledge our lab mates, lab members. First one is that Kanta Ashok as well as Subhash Chandra. So both of them recently defended their thesis, and remaining 2 or 3 is under pipeline. One is Banat Karan, as he is currently pursuing, and one member is in off campus, and that other last guy is Fahim. So he did our MPharm thesis. So these are all our lab mates. And finally, I would like to thank the entire organizing team, that is organizing chair Dr. NVR Naidu as well as Dr. V. Madhavan, as well as organizing co-chair, that is Bindu Yes, as well as Venkatramana, as well as our coordinators, that is Krishna Murthy, as well as Dr. P. Parashuraman, as well as Maniyanta Murahari for Department of Pharmaceutical Chemistry, this Ramaiah Institute of Applied Sciences, as well as Ramaiah Institute of Technology, for their wonderful opportunity to share our research findings in this wonderful forum. So thank you. Thanks a lot. Finally, I would like to thank our management also, as well as our vice chancellor, as well as campus director, as well as HOD, as well as all my departmental colleagues, in order to make this presentation in a successful manner. Yeah, thank you. Thank you, sir. Thank you.
Yes, sir. Thank you so much, sir. Thank you so much for sharing your wonderful knowledge, wonderful experience, what you had in the drug discovery process, that too in drug repurposing. And here we have some questions from our audience sides.
Yeah, please.
So we'll move on to question session.
Yeah, please.
So, are hidden Markov models used for virtual screening and scoring? Hidden Markov models.
Can you repeat? Can you repeat?
Are hidden Markov models.
Okay. Okay.
Used for virtual screening and scoring?
Yes, yes.
Fine. So what is the difference between hit identification and the lead optimization?
So hit identification means it's a preliminary or raw data kind of thing. So which may not obey your Lipinski rule of 5 or Lipinski rule of 3 kind of thing. So that we can consider as a hit. Then lead means it's a previous, just one step again to the drug. So if it obeys all your Lipinski rule of 5 or 3 or lead like filters, lead like parameters, everything is obeyed, then it will be considered as a lead.
And the next one is for a repurposed drug. Its ADME is already known. So what is the first step or approach in assigning that drug as the repurposed one? Sir, I will repeat again.
Yeah, please.
For repurposed drug, its ADMEs are already known.
Okay.
So what is the first step or approach in assigning that the drug is repurposed one? From which step we should start? So example, if a drug is that for the drug, how we have to start? Whether it is a repurposed one or whether we can repurpose, like that they have raised the question.
So in that case, if we apply that ADMET parameter, sometimes some exceptional drug, for example, if you take a Taxol or paclitaxel,
yes.
So it is an exceptional case. So if you apply any filter, definitely it will fail. Okay, so but it will be added in your that database as a drug.
Okay, sir.
Okay, so that kind of molecule, if we apply, then we will lose that. So it will not get filtered out.
Fine, sir.
Okay, so in that case, if it is of all FDA approved drugs, then normally all those filters may not be required.
Okay.
But sometimes if that database containing a mix of FDA approved drugs as well as new chemical entity, then that ADMET filters is must.
Fine, sir. Yeah. So how you done standard precision for your work? How you have done standard precision for your work?
So it's a module available in Glide. XP is the first standard precision, then followed by extra precision.
Fine, sir. Fine, sir. So what is co-crystallization? How to run co-crystallization in docking?
No, so first what we have to do, so we have to download the PDP from protein data bank. Then from that we need to exactly pinpoint which is co-crystallized ligand. Okay, then that co-crystallized ligand we need to select, and the extraction option is there. Extract.
Okay, sir.
Just we can simply remove that co-crystallized ligand from the active site.
Sir.
Then that extracted co-crystallized ligand, we have to assume that is a new ligand. Then we need to prepare that co-crystallized ligand. Then we need to perform docking for that.
Okay,
so we
okay,
get the docking score as well as the docked pose. Then again we need to superimpose that extracted ligand on top of the docked pose.
Fine, sir.
Yeah. So this is the way.
So one more question.
Yeah.
From a person. I performed docking protein ligand using Autodock Vina.
Okay.
PL and TS glide, and I repeated the process for 3 times for each ligand.
Okay.
So I got different binding energy each and every time. Why I am getting different results from same software in every step?
Yeah. Correct.
So which step should docking should consider, or I should take average binding score?
That will be always better. Average. So
average only. So
it will not vary much. No. For example, first time -14 you may got. Second time -13.9 or 13.8 you may get. And third time 14.20 something you may get. Likewise you got, or -14, -10, -5, something like that you got.
Fine.
How you got? 3 times you have performed.
Okay, sir. So how the pharmacokinetic property of a lead molecule against a specific target was assessed to the drug likeness?
So no, it is not anything specific to the target.
Okay.
It is specific to the ligand, what we have identified. So our body is being considered as a system. Okay, so that system, if you are introducing any new chemical moiety, means how it will interact or how it will behave. So that thing we are predicting. So it is not anything specific to the target. Yes.
Okay, sir. Can we repurpose any drug, or is there any selection strategy?
Selection strategy in the sense either we can
any hypothesis we have to create, or is like
that will be ideal always.
Okay, sir.
So without that, then finally justification will become very difficult. Yes.
Fine, sir. After the molecular docking study, how would I know whether the binded ligand is agonist or antagonist?
Okay, very good question. By means of a docking study alone, we may not be able to differentiate that.
Okay.
So for that, we need to perform experimental study only.
Okay.
Dose response curve or this thing only we need to perform.
Fine, sir. So how to get radius of gyration for protein, upper protein in this month? Did it generate on its own?
No, we can define it.
Okay, sir.
Yeah.
So which one is better to get appropriate drug target, drug repurposing, or for drug target, drug repurposing or reverse vaccinology? Which is the best method? Either drug repurposing or reverse vaccinology?
Reverse?
Vaccinology. Vaccinology.
I'm not sure about reverse vaccination.
Okay, sir. Can you explain the importance of protein ligand Rmsd and protein Rmsf in molecular dynamics?
So Rmsd is nothing but root mean square deviation. So it indicates how much any specific amino acid residues involved in the binding is deviating.
Okay, fine.
Then Rmsf, in the sense, it is a fluctuation. So here, in that we will consider only fluctuation, how it is fluctuating during the entire time limit what we have studied.
Okay, sir.
5 nanoseconds. Out of 5 nanoseconds, how many nanoseconds it was fluctuated and how many nanoseconds it was static. So that thing we will be calculating.
Okay, sir. Okay, sir. That's all, sir. Thank you so much for sharing your knowledge, sir, and you have reduced our burden, because tomorrow for molecular docking hands-on training, you have given a brief introduction. What is the docking and what are the requirements, why we have to do.
Okay.
So we are very happy to have you as a speaker, sir. Thank you so much for your presentation.